Now I would like to invite Dr. Fatima Muccee, Assistant Professor, Department of Biotechnology, Virtual University of Pakistan, to please come and enlighten us.
Assalamu alaikum. I have been introduced, and now I'm going to present my topic entitled Whole Genome Shotgun Sequencing of POPs Metabolizing Bacteria Isolated from Tannery Industry Effluents and Petrol Contaminated Soil. Great. First of all, I will introduce the POPs. POPs stands for persistent organic pollutants. These are highly toxic synthetic carbon-based compounds which are persistent. They tend to bioaccumulate in organisms and they exhibit a long range of transport. They have wide range of hazardous effects on the ecosystem as well as the human health. Their health hazards include insulin resistance, thyroid disorders, obesity, decreased immunity, endocrine disruption in early age, cancer, damage to central nervous system, reproductive problems, birth defects, and death. So keeping in view all these hazards caused by POPs, a global treaty was led by the United Nations Environment Programme to protect human health and environment by reducing or eliminating the production, use and release of initially categorized 12 key POPs, which were termed as the dirty dozen, which includes the pesticides, industrial chemicals and unintentionally produced byproducts. So for various multiple conventional approaches have been employed for POPs mitigation, like soil vapor extraction, incineration, ultraviolet oxidation, nanotech remediation, and chemical oxidation. But these suffer from wide range of disadvantages, like they are high cost, they involve incomplete degradation of POPs, they cause additional environmental risks, they need transportation of contaminated material, they cause enhanced exposure to pollutants for workers, and they involve more complex technology. So, what can be the best possible solution to deal with such hazardous compounds like POPs? It's a new approach, which is bioremediation. Bioremediation is degradation of environmental organic pollutants by plants, fungi, or bacteria, which are capable to consume and metabolize the contaminants and thus neutralizing the environment. The bioremediation has various advantages as compared to the conventional approaches. Like it is a low-cost technique. It involves it completely removes the POPs. It is eco-friendly, cause minimum disturbance to operation site. It detoxifies environment without additional risk. It involves no need of transportation. Like it's an in situ technique, and in most of the cases it avoids exposure to pollutants, and it involves simpler technology. In literature, we found various studies that have analyzed and various POPs metabolizing bacteria using the metagenomic approaches like the bacteria belonging to genus D. sulfuromonas, which is capable of metabolizing toluene, benzoate, paracresol, 4-hydroxybenzoate, and phenyl acetate, has been analyzed using Illumina sequencing of metagenomic DNA labeled with C13. Similarly, another bacterium, Rhodococcus strain, which is capable of metabolizing biphenyl, has been analyzed through whole genome shotgun Illumina sequencing. And the bacteria belonging to Clostridiaceae and D-sulfuromonodeals, which is capable of naphthalene metabolism, has been characterized through DNA-based stable isotope probing and metagenomic analysis, and a beta proteobacteria, which is capable of degrading polycyclic aromatic hydrocarbons has been subjected to DNA-based stable isotope probing and metagenomic analysis. So keeping in view all these literature, all these findings in literature, and also the hazardous effects of the persistent organic pollutants, we hypothesized in this study that through whole genome shotgun sequencing of POPs metabolizing bacteria, which we have been isolated from different native sources, we might find some novel genes and pathways associated with the POPs degradation. Before introducing my experimental strategy, let me tell you that I have isolated in this project already isolated 41 POPs metabolizing bacteria, of which 8 were petrol metabolizing, 28 were toluene metabolizing, and the remaining were capable of degradation of naphthalene. And all these were characterized through next generation sequencing. And wa- we uh, we performed 16S ribosomal RNA metagenome sequencing as well as whole genome shotgun sequencing. For this purpose, all the 41 bacteria were subjected to the process of DNA extraction by organic method. And after the extraction of DNA from each individual isolate, I constructed a DNA consortium. And this DNA consortium comprised of the equimolar concentration of DNA from each of the individual isolates. And this DNA consortium was then sent for Illumina sequencing in Macrogen, Korea, where it was subjected to quality control analysis. The QC analysis of DNA of post metabolizing bacterial consortium showed their concentration of approximately 34 nanograms per microliter, and the presence of DNA was further, and the quality of DNA was further confirmed by running it on agarose gel electrophoresis, and we found a band of size 4,000 base pairs and comprising of 40 nanograms of DNA per 5 microliter. So after ensuring the quality of the DNA, it was subjected to the library construction for 16S ribosomal RNA metagenome sequencing. We used the library kit of Hercules 2 Fusion DNA polymerase Nextera XT index kit V2, and followed the protocol of 16S metagenomic sequencing library preparation. While for whole genome shotgun sequencing, we used the kit library kit of 2C DNA polymerase DNA PCR3 was prepared following the protocol True-seq DNA PCR-free sample preparation guide. It was followed by the PCR amplification, and for 16S ribosomal RNA, we targeted the region of the gene which was between variable 3 and variable 4, and the forward and the reverse primers used are also shown here, and the PCR program which was used for this purpose is also shown in detail. Next step was sequencing. For 16S ribosomal RNA sequencing, metagenome sequencing we used the platform of Illumina HiSeq 300 base pair paired and 30% X sequencing platform. While for shortened whole genome sequencing, we used HiSeq 1100 base pair paired and per sample sequencing platform. The data analysis. The sequencing data was converted into raw data through Illumina sequencer using sequencing control software for system control. The real-time analysis was used for base calling. Illumina package BCL to FastQ was used to convert BCL base calls binary into FastQ. Trimomatic version 0.36 software was used to trim adopters from 16S ribosomal RNA gene, and shotgun sequencing reads software. For this purpose, mainland Illumina clef and sliding window size were kept constant. The raw data we got comprised of forward and reverse sequencing files. Next step was metagenome assembly. For this purpose, MegaHit metagenome assembler with different ranges of k-mer sizes, that is 29 to 119, was used. for assembly of NGS reads in contigs with less than 500 base pairs were filtered to carry out metagenome annotation. The cost quality parameters are discussed here in detail. You can see the total number of contigs, the number of contigs which are less than 1,000 base pairs, and number of contigs more than 5,000 base pairs, number of contigs more than 10,000 base pairs, number of contigs more than 25,000 base pairs, and number of contigs more than 15,000 base pairs, and GC percentage. All the cost quality parameters are mentioned in detail. Next, the bioinformatic pipeline, MG-ROST, that is metagenomic rapid annotations using subsystems technology, was used for the taxonomic characterization, and filtered 16S ribosomal RNA gene contigs were aligned. Assembled contigs were subjected to MG-ROST pipeline using default QC parameters like 60% minimum identity cutoff and 15 base pair minimum alignment length, and so on. The BLAT algorithm was used to identify using Euclid's algorithm. The reads generated were then subjected to taxonomic analysis using NCBI RefSeq database. For here, the annotation of contigs against selecting databases at different BLAT E values is shown here. Now, for de novo annotation of assembled shotgun sequencing contigs, the coding sequences were predicted using a metagene program. The predicted coding DNA sequences were annotated against different databases like seed subsystems, KPGG, and COG databases were selected for functional annotation of contigs. To perform functional annotation using these databases, we used MG-ROST server, and sequence streets obtained were deposited in NCBI sequence street archives under the accession numbers, which are shown here. What we got after the taxonomic profiling, the taxonomic profiling of contig dataset is represented here in the form of a corona chart, and each circle of the chart represents a subsequent level of taxonomy from domain to genus level, and bacterial sequences were dominant on domain level, with more than 99% of the contigs annotated against bacterial sequences. On phylum level, firmicutes demonstrated themselves to be the most abundant with the 80% abundance, followed by the proteobacteria with 12% abundance, and then actinobacteria with 5% abundance, which was and on genus level. Bacillus was found to be the most abundant with the 45% abundance, followed by Burkholderia with 25% abundance, and then Brevibacillus with 9%, and then Geobacillus with 4% abundance. The functional profiling. Actually, we performed the whole genome shotgun sequencing for two purposes. One was to identify the genes in all the 41, in the DNA of all the 41 bacteria, which might be associated with the metabolism of any of the aromatic compounds which are considered as POPs. But as you know, in whole genome shotgun sequencing, we can get all the genes that can be associated with any other kind of metabolism or any other kind of function. So metabolic pathways against with the genes were annotated is shown here with the respective abundance. Like you can see, the most number of genes were annotated against the carbohydrate metabolism, that is almost 11,000, and followed by the amino acid metabolism, which is 800, and then for protein metabolism, which is 500, and you can see here that the almost approximately 200 genes were found to be associated with the metabolism of aromatic compounds. The next, we also tried to identify all the possible pathways which can be found in these 41 POPs metabolizing bacteria, and our results are shown here. The most number of, you can see here in this figure, the benzoate degradation. The most number of genes were found to be associated with the benzoate metabolism, that is 13%, and then with the phenyl propanoate compound degradation, that is 11%, and then with homogenizate pathway degradation, in 10%, and so on. Actually, the bacteria we isolated were capable of metabolizing the toluene, naphthalene, and petrol, and different components of petrol. But when we subjected them to shotgun whole genome sequencing, then we came to know that they have a diverse potential of metabolism of wide range of POPs. Here, the genes which have been identified through different subsystems are shown. Like, for example, from KEGG functional annotation, we were able to identify the genes encoding phthalate, 4,5 dioxygenase enzyme, salicylate hydroxylase enzyme, and so on. While through seed subsystems functional annotation, we were able to identify almost 66 and genes encoding enzymes that might be associated with the degradation of different kinds of POPs, as you can see here, like benzoate, biphenyl, cinnamic acid, chloroaromatic compounds, and phenyl propanoid, catechol or salicylate, gentizate, para-hydroxybenzoate, toluene. And on the basis of identification of these genes, then we also identified an early reported pathway of toluene metabolism in the present study bacterial consortium. And in this pathway, you can see the toluene is through multiple reactions is ultimately converted into acetyl coenzyme. You know, the acetyl coenzyme is a part of the major central carbon metabolism process through which the bacteria can, or any other organism can get the energy for their body activities. So it means that these bacteria are capable of metabolizing toluene and using it for the growth and other activities of the body. The enzymes which we identified from this pathway through whole genome shotgun sequencing are highlighted in red, like benzyl alcohol dehydrogenase, benzoate 1,2-dioxygenase, 2-oxopen 4-enoate hydratase, 4-hydroxy 2-oxovalerate aldolase, and acylating aldehyde dehydrogenase. Next, we were also able to identify the pathways which have been reported in earlier different POPs metabolizing bacteria these pathways might be associated with the procatechate and catechol degradation. Like you can see here, the phenol and ethyl benzene, they can be converted, they can be broken down. They can be broken down into they can be broken down through this mechanism into catechol, and through they are broken down into catechol. Actually, phenol, either it is ethyl benzene or any other alcohol or toluene, it is ultimately converted into catechol, and then catechol is further processed through two pathways. Like, one is the beta ketoadipate pathway, and the other is the procatechate pathway. So we were able to identify the enzymes of both these pathways, the procatechate branch and catechate branch of beta ketoadipate pathway. Hence, the whole genome shotgun sequencing helped us a lot in identifying the pathways which can be which we can suggest to operate in the bacterial consortium, and which can be helpful for us in future for the metabolism and remediation of the POPs. And some of the other enzymes that have been identified in these POPs through the NGS analysis and the reaction catalyzed by them is shown, like the enzyme associated with vanillate degradation, the enzyme associated with salicylate, benzaldehyde, phthalate, cyclohexanone degradation. Hence, through this study, we can conclude that actually we isolated the 41 bacteria from Tannery effluents and Petrol contaminated soil. So these two resources were inhabited by a diverse array of bacteria exhibiting bioremediation potential for wide variety of POPs, and approximately 200 genes were found to be associated with degradation of POPs, and some of the virulence genes were also identified through NGS analysis in these bacteria, and some of these bacteria were also found to exhibit the antibiotic sensitivity, to exhibit the antibiotic resistance. So we cannot recommend the whole bacterium. Therefore, we cannot recommend the whole bacterium to be used for eco-friendly bioremediation of POPs. However, still these bacteria are very much useful for us because we can identify, we can isolate their genes, we can cut them from these bacteria, and we can insert them in the eco-friendly expression systems and can utilize them for POPs remediation. In the same way, we can identify, we can isolate or extract the enzymes from these bacteria and can utilize them in eco-friendly way for the bioremediation purpose. And this work is from my PhD project that has been published in an international journal of microbiological research with impact factor 3.7. Thank you so much.
You're welcome, Dr. Fatima. And we have a question from the audience, if you will, please.
Yes, please.
Okay.
Nice presentation, ma'am Fatima. I have a question for you. What type of online softwares are used by yourself for the analysis of whole genome sequences?
I have mentioned all the online, all the tools and all the pipeline in the study which have been used for this purpose.
Yeah, she did Omnix box and everything. Okay, thank you, Dr.
Yeah, I have a quick question, Dr. Wasim here from ComSets.
Yes, Dr. Wasim, please proceed.
Ji, well done, Dr. Muccee. Excellent work. I'm sorry, how do you pronounce your name, Dr. Fatima?
Ji.
Yeah, maybe I pronounced it correctly. Where did you work on, especially this sequencing thing? From where you get your sequencing thing done?
Sorry, sir. Can you repeat your question? I'm not able.
From where did you get your sequencing done, the NGS thing done?
I sequenced them from Macrogen, which is in South Korea.
So through some local, sequencing company or something?
No, it's not a local one. And I will recommend you to send your sample to Macrogen, South Korea, and never to any local company.
Okay. And have you, I'm sorry, I missed the first part probably. So how did you do your sampling? These were water samples?
No, actually, I selected the Tannery industry effluent for the isolation of naphthalene and toluene metabolizing bacteria, and I took petrol contaminated soil from the gasoline station or petrol pumps, and I isolated petrol metabolizing bacteria from that source.
Okay, good work. Thank you so much.
Thank you so much.
Thank you, Dr. Fatima.
Thank you so much.
That was an excellent presentation.